Hello guys, welcome back to Molecular Biology and Biotechnology with Lucy. Today we'll be talking about the 6 steps of recombinant DNA technology. Recombinant DNA technology allows us to produce DNA via artificial means through combination of genetic material from different sources. Now, these new genetic combinations are of value to science, medicine, agriculture, and industrial applications. So let us look at the 6 steps. Number 1, isolate the vector and the target gene. So in this case, we are saying we want to have 2 DNA sequences being combined. So we have what we call a vector gene and a target gene. So this is probably what you want to propagate, and the vector is what is going to hold our target gene. For example, we are going to use a prokaryotic bacteria cell and a eukaryotic animal cell. So our bacterium, we are going to extract plasmid. We talked about plasmids in the structure of the cell. A plasmid is an extrachromosomal DNA in prokaryotes, and this is the chromosomal DNA in bacteria. So this is extrachromosomal, usually double-stranded circular DNA. And we want to also now extract our target gene, which we are going to get from the chromosomal DNA of our animal cell. So we have extracted. I'm going to link the extraction of DNA in this video, and I will also be doing another video later on the extraction of plasmid, the step-by-step protocol of extraction of plasmid. So now we have isolated the plasmid DNA and the DNA that contains our gene of interest. Next, step number 2, cleavage with restriction enzymes. So we have now our plasmid, which is circular, and we have our very long DNA, which we need to get the gene of interest. So we get a restriction enzyme, which we'll be looking at in more details, but these enzymes usually cut at specific locations, and we cut, you can see here, we cut our plasmid so that we can remove a section, usually at a cloning site. And then we also use the same restriction enzyme to cut our chromosomal DNA so that we can get that fragment of interest. Step number 3, join or ligate into the vector. So now we have our plasmid DNA, which we already cut and removed this section. So the section of DNA that is our interest, now you insert into the vector, and this one usually we use an enzyme called ligase enzyme. Step number 4, transformation into the host. Now a plasmid cannot propagate on its own. It needs to go back to a bacterial cell. So we need to do what we call transformation by putting a plasmid into a bacterial cell. So this is a recombinant plasmid. We need to put it into a host, which is a bacterial cell. That is what we call transformation. And this is how now our recombinant bacterium will look like. Number 5. Now, it is possible that if you have several bacterial cells, some of them are able to take up the recombinant plasmid and others are not able to take up the plasmid that is recombinant, and therefore they will not have become recombinant. So how do you know, or how do you select those that have picked up our recombinant plasmid and differentiate them from those that did not? We perform what we call screening or selecting. And here we usually use selectable markers. For example, there is a marker called antibiotic resistance, let's say to ampicillin. So it's a resistant gene, or streptomycin resistance. So these genes usually will be found on the plasmid, here on the plasmid. So once you clone, or once you have put your insert, our resistant gene is probably somewhere there. Let's use for ampicillin in this case of this bacterial cell. So what is going to happen is, because it has a resistant gene for ampicillin, then if you grow our cells in antibiotic containing medium, okay, so this is an antibiotic containing medium here, which has now ampicillin antibiotic, then it means that those that were able to pick up the plasmid that is recombinant are going to grow, because they already have a resistant gene against this antibiotic. Those that did not take up our plasmid will mean they will die, because they do not have a gene that is resistant to ampicillin, so ampicillin destroys them, and that is what we call screening. Or in our medium, only those that have recombinant plasmid grow. Okay, that is step number 5. Step number 6. Why are we doing all this? We said that the combinations are useful to science, agriculture, medicine, and so on. So for what reason is now where we have step number 6. Mainly, we have cloning, and cloning means many copies, making many copies of something. So you probably just want to propagate your gene of interest. Remember the small gene of interest here, the fragment gene of interest. You probably just want to propagate. So you clone, you grow as many cells. Remember we talked about subculturing in our previous lesson. So you want to grow and make as many copies, and from there you could extract. So this is somehow the traditional method, which is now being called PCR, or polymerase chain reaction, where we are able to multiply fragments of DNA or genetic material to have as many copies for tests in our laboratories and so on. So traditionally, because PCR was not there, then we were using culture in cloning or making many copies of. There is also another use where now we want to express proteins. So probably your gene of interest that we have inside this plasmid is for producing a certain protein, let's say insulin for diabetic patients. So you will have it, and as you make many copies, it means that the process of translation is also happening, and as you do translation, it means that you're producing insulin. Okay, so there are many applications of this recombinant DNA technology, which we have talked about some of them. Production of proteins in medicine. We have GMOs. You can make, plants that are pest resistant. You can also use them in environment where you select a gene so that you alter bacteria so that they're able to clean up toxic waste, and so many other applications. So those are the 6 steps of recombinant DNA technology. That's it, guys. We shall be doing a more detail in the 6 steps, but for now, if you have any questions, don't forget you can inbox, you can write on this wall, you can write me an email. Use as many ways as are available to you to be able to reach me. Thank you so much. See you in our next lesson.